Welcome back to G Fun Facts Online. Today, we are diving into a subject that fundamentally rewrote the textbooks on how the human brain operates. We are going to explore the sub-millisecond world of neurotransmitter release. For a long time, this process was considered the dark matter of cellular biology. It is a mechanism so fast and occurring on a scale so small that for decades it existed almost entirely in the realm of theoretical inference and electrophysiological squiggles on a screen. To understand the magnitude of the breakthrough we are discussing today, we have to look at the history of neuroscience. For decades, neuroscientists were essentially like astronomers trying to understand the birth of a star, but only having access to a static photograph taken millions of years after the event occurred. Through traditional imaging, they could see the before, which were synaptic vesicles docked at the membrane, and they could see the after, which were empty vesicles and fused membranes. But the explosive, dynamic middle, the actual event of communication, remained invisible. That all changed with the advent of a technique called Zap-and-Freeze electron microscopy, also known as flash and freeze. This revolutionary technique was pioneered by researchers like Shigeki Watanabe and Eric Jorgensen, and it effectively broke the speed limit of biological imaging. By coupling electrical or optogenetic stimulation with high-pressure freezing, scientists can now freeze a neuron in the exact midst of firing. This captures the precise moment a synaptic vesicle fuses, collapses, and is recycled, all with millisecond temporal resolution and nanometer spatial resolution. In this episode, we are going to explore the mechanics, the discoveries, the controversies, and the future of Zap-and-Freeze. To appreciate why this is such a massive leap forward, we first need to understand the static problem of traditional electron microscopy, or EM. Since the 1950s, EM has been the gold standard for visualizing the ultra-small. It revealed the existence of the synaptic cleft, the dense clusters of vesicles, and the dark active zones where release occurs. However, traditional EM has a fatal flaw, and that flaw is time. Preparing a sample for standard EM is typically a slow and somewhat violent process. Tissue is usually soaked in chemical fixatives like glutaraldehyde and osmium tetroxide. These chemicals take seconds to minutes to diffuse into the tissue and cross-link the proteins to preserve them. In the world of synaptic transmission, an event that lasts less than a millisecond, a duration of seconds is an eternity. Chemical fixation effectively captures a dead state, often filled with artifacts where membranes blister, organelles swell, and fast-acting proteins relax into non-native conformations. In the 1970s, researchers John Heiser and Tom Rees attempted to solve this with a technique called slam freezing. They literally dropped tissue onto a block of liquid nitrogen-cooled copper. The impact froze the surface layer instantly, physically trapping structures in place without chemicals. This allowed them to see vesicle fusion for the first time, which was a landmark moment. But slam freezing had severe limits. It only preserved the very surface, about 10 microns deep, and mechanically distorted the sample because, as the name suggests, the slam was literal. Furthermore, synchronizing the freeze with a neuronal stimulus was incredibly difficult. They could see that fusion happened, but the precise, millisecond by millisecond choreography remained blurry. This brings us to the mechanics of Zap-and-Freeze. This technology is not just a better camera. It is effectively a time machine. It combines three sophisticated technologies: precise stimulation known as the zap, high-pressure freezing, or the freeze, and robotic synchronization. Let's break down the zap first. The core innovation is the ability to trigger a neuron to fire exactly when you want it to. A custom-built field stimulation device is inserted directly into the freezing chamber. It delivers a precise 1 millisecond electrical pulse, typically 10 volts per centimeter, to the tissue. This mimics the natural action potential of a neuron, causing all chemically excitable synapses to fire simultaneously. There is also a variation called flash and freeze, where neurons are genetically modified to express channel rhodopsin, a light-sensitive ion channel. In this case, a flash of blue light triggers the neurons. This optogenetic approach is less invasive than electricity and allows for specific targeting of certain neuron types, such as activating only inhibitory neurons in a mixed culture. Next, we have the freeze. Freezing biological tissue is incredibly tricky. If water freezes slowly, it forms sharp hexagonal ice crystals. These crystals puncture membranes and shred the cell, causing what is known as ice damage. To prevent this, the Zap-and-Freeze method blasts the sample with a jet of liquid nitrogen at 2100 bar, that is, 2100 atmospheres of pressure. At this immense pressure, the physics of water changes. The water enters a vitrified state. It becomes a solid, amorphous glass without forming ice crystals. This preserves the biological structure in its native, liquid-like arrangement, but halts all molecular motion instantly. This freezing process happens in approximately 8 milliseconds. That is the shutter speed of our camera. Any biological process happening slower than 8 milliseconds is effectively frozen in time. The third component is the synchronization. The magic lies in the timing. The system is controlled by a computer that coordinates the zap and the freeze with microsecond precision. At time 0, the computer sends the zap stimulus. At 5 milliseconds, the sample acts as if it is alive. Vesicles fuse and neurotransmitters release. At 10 milliseconds, the high-pressure jet hits and the sample is frozen. The sample is now a permanent snapshot of what was happening exactly 10 milliseconds post-stimulus. By repeating this experiment with different delays, freezing at 15 milliseconds, 30 milliseconds, 50 milliseconds, 100 milliseconds, and 1 second, researchers can build a flipbook animation of the synaptic cycle. The most significant discovery made with Zap-and-Freeze, the one that shook the foundations of cellular neuroscience, was the identification of ultrafast endocytosis. For 40 years, the textbook model of vesicle recycling was clathrin-mediated endocytosis. The theory was that a vesicle fuses and collapses fully, the membrane drifts to the side of the synapse, and then the protein clathrin slowly assembles a soccer ball-like cage around the membrane to pinch it off. The time required for this was thought to be 20 to 60 seconds. This presented a mathematical problem. Neurons can fire at 100 Hz, or 100 times a second. If recycling took 20 seconds, the nerve terminal would run out of membrane in seconds. The math simply didn't add up. In 2013, using Zap-and-Freeze on mouse hippocampal neurons, Shigeki Watanabe and Eric Jorgensen looked at what happened in the gap that traditional EM missed, the first 100 milliseconds. What they found was shocking. At 30 milliseconds, vesicles were fully collapsed into the membrane. Between 50 and 100 milliseconds, huge invaginations of membrane appeared at the edges of the active zone. By 100 milliseconds and beyond, these invaginations pinched off into large, irregular vesicles about 4 times the size of a normal synaptic vesicle. This process was happening in less than 1/10 of a second. That is 200 times faster than clathrin-mediated endocytosis. They named it ultrafast endocytosis. Subsequent studies revealed the machinery behind this speed. The fast invaginations were uncoded, meaning clathrin was too slow to be involved. The membrane was being pulled inward by actin filaments, the cell's internal muscle. Furthermore, the protein dynamin, which acts like a pair of scissors to cut the vesicle neck, was already pre-positioned at the edge of the active zone, waiting for the signal to snip. This discovery solved the membrane budget crisis. The neuron wasn't carefully rebuilding vesicles one by one during high activity. It was frantically grabbing large chunks of membrane to clear the active zone for the next round of firing, sorting them out later in the endosome. Science is rarely a straight line, and Zap-and-Freeze landed right in the middle of one of neuroscience's most heated debates, the kiss-and-run versus full collapse controversy. For decades, a faction of neuroscientists argued that full fusion, where the vesicle flattens completely, was too slow and wasteful. They proposed the kiss-and-run hypothesis. In this model, the vesicle touches the membrane for a kiss. A tiny pore opens to let neurotransmitters out, and then the pore closes immediately as the vesicle detaches and runs. The advantage here is extremely fast recycling with no need to rebuild the vesicle. Electrophysiological data seem to support these transient pores. However, when Watanabe and colleagues used Zap-and-Freeze to look for these events, they saw vesicles collapsing completely into the membrane within 30 milliseconds. They did not see vesicles retaining their shape at the active zone after fusion. Their data suggested that at physiological temperatures and normal stimulation, full collapse followed by ultrafast endocytosis is the dominant mode. Kiss and run might be rare or specific to certain cell types or stress conditions. However, recent advancements from 2023 to 2025 using cryo-electron tomography combined with Zap-and-Freeze have added nuance, leading to a unified kiss-shrink-run model. Researchers observed that vesicles open a pore, rapidly lose volume and surface area as lipids diffuse out or the membrane remodels, effectively shrinking, and then they are retrieved. This suggests the dichotomy might be false. The full collapse might just be a very deep shrink that looks like a flat membrane in 2D. But 3D tomography reveals a more complex topology. Why does all of this matter clinically? Zap-and-Freeze is beginning to explain human disease. Take epilepsy and seizures, which are characterized by uncontrolled neuronal firing. Studies have shown that the bottleneck of synaptic transmission is often the recycling machinery, specifically dynamin. If ultrafast endocytosis fails, the synapse jams. Vesicles cannot be cleared, and the neuron enters a state of depolarization block or erratic firing. Understanding the millisecond timing of this failure helps in designing drugs that target the recycling machinery rather than just the receptors. In Parkinson's disease, recent work in 2025 applied Zap-and-Freeze to human brain tissue derived from neurosurgery. The findings showed that the mechanism of ultrafast endocytosis is conserved in humans. Several genes implicated in Parkinson's, like LRAK2 and endophyllin A, are directly involved in the membrane curvature and scission steps of ultrafast endocytosis. The hypothesis is that in early Parkinson's, traffic jams at the synapse due to slow recycling might precede cell death. This technology allows researchers to see these jams in animal models before any symptoms appear. It is also crucial for hearing and vision. Sensory neurons in the ears and eyes must fire continuously at incredible rates, up to 500 Hz. Zap-and-Freeze has revealed specialized ribbon synapses in these organs that use a unique variation of ultrafast recycling to handle this relentless throughput. Looking to the future, the field is moving from 2D snapshots to 3D movies. The next frontier is combining Zap-and-Freeze with cryo-electron tomography, or cryo-ET. Standard Zap-and-Freeze takes a 2D slice. Zap-and-Freeze ET takes a frozen sample, tilts it at different angles, and reconstructs a 3D model of the synapse at the single molecule level. The vision is that researchers can now see individual connector proteins appearing and disappearing in 3D space milliseconds after an action potential. We are no longer just seeing the vesicle. We are seeing the engine that drives it. Scientists are also combining this with super-resolution correlative light and electron microscopy, or CLEM. This involves flashing the neuron to stimulate it, freezing it, using a super-resolution laser to identify specific proteins, and then using EM to see the membrane structure. By overlaying the two images, scientists can tag the invisible actors in the electron micrograph, confirming exactly which proteins are driving the membrane bending. In conclusion, Zap-and-Freeze technology has fundamentally transformed our understanding of the brain's most basic unit of currency, the synaptic vesicle. It turned a static field into a dynamic one, revealing that the synapse is a place of furious, violent speed, where membranes collapse, explode, and regenerate in the blink of an eye. By capturing the ghosts of intermediate states that exist for only a few milliseconds, Zap-and-Freeze has solved the mystery of how our brains can think faster than our cells can seemingly build the machinery to support it. As we push this technology further into 3D and human pathology, we are likely to find that the milliseconds we ignored were actually the most important moments of all. Thank you for listening to Fun Facts Online.